I'm David Wohl here at the University of North Carolina, and with me is Joe Iran, the PI of our AIDS clinical trials unit here, and Christopher Hurt, who is at the end there and specializes in many things, including HIV prevention and hepatitis C and influenza and general ID. Good. So why don't we start off with Ada?
So I just wanted, those were great presentations. Thanks a lot. And Christopher, I enjoyed yours also. I just want to make sure something.
Sure. So what Ada commented on is, it's a great point, is that the women that were participants in ISIS were specifically selected and recruited because they were at high risk for HIV infection. So the venue-based sampling and then individual and partner-level characteristics were part of the selection process for participating. So that incidence estimate doesn't reflect all black women in the United States. It's more generalizable to women that are at highest risk. So women that themselves lived in an area that has a high prevalence, had partner or individual characteristics that are similar to the women in ISIS. The same is true for brothers, that it's not true for all black MSM in the United States. It's more generalizable to those men who fit the characteristics that are similar to the participants in the Brothers study or 061.
And just to follow up to that,
yeah.
And the reason for that perhaps somewhat sort of idiosyncratic sort of recruitment method was because one of the original purposes of the study was to demonstrate the feasibility or determine the feasibility of doing an HIV seroincidence study among women in the United States, given the fact that the incidence is relatively low.
Yeah.
So I mean, I wasn't in charge of how ISIS was presented, and I don't think either of you two were either. I think it was completely mispresented, if you want my opinion, which I guess you're going to get whether you want it or not. Which is, to me, it was stunning how low the incidence was, and I think what ISIS told me was how hard it is to find women that are at high risk. We see women all the time that are newly infected with HIV, yet out of 2,000 women, or 2,000 plus women, who were specifically recruited from high incidence areas and all those other things that Christopher and Dr. Adamora said, there were 4 seroconversions total. You won't be able to do a study of a prevention in that population because it will be statistically impossible. Yet we know women are getting infected. So to me, it was a completely different story than this, 5 times higher or 10 times higher than the CDC estimate, which I think was not the way to go, personally. And to me, the Brothers study and the ISIS study are really very contrasting. I mean, we can do prevention studies in young black MSM because they have a 5% incidence. We can do that. How are we going to do prevention studies in women? I mean, I throw that question back to you, Dr. Adamora, because I don't know how we're going to do them.
But I think that actually, I agree with you. I think that's a really critical point. And it creates a huge problem for women in the United States. I mean, yes, the incidence was well, it depends. The glass is half full, the glass is half empty, how you decide to look at it. Among those women, that is in some ways a relatively high incidence, but the most critical point is that it is still too low to do incidence studies in the United States that have HIV as an outcome, and that's essentially what you need to have to demonstrate efficacy of whatever intervention it is you're trying to test. And that is an extraordinary problem because those interventions that work elsewhere may not necessarily work here in the U.S. And that is a huge methodological and therefore clinical problem. I agree completely.
All right, other questions? Kate?
Yeah, David, I just wanted to...
Yeah, so what Kate's bringing up is that with single tablet regimens, with these formulations, are there particular advantages vis-a-vis adherence? and especially selective adherence, where if you have 3 different types of medication that you're taking, even if it's once a day, you may adhere to 2 of them, but you don't like the yellow pill for some reason, or you forget to take it. And that could lead to really a good setup for bad things to happen to you, like resistance, virologic breakthrough. So, there are emerging data that you can imagine who's really interested in producing and propagating those data. Read the people who make single tablet regimens. But there's a truth lining that. And I remember when we went from, looking at twice a day to once a day, and there was some pushback from people who were really vested in twice a day therapy. Does twice a day really make that much of a difference compared to once a day? And there's certainly a lot of literature outside of HIV about dosing frequency and pill numbers and pill size that we can learn from. But even within the realm of HIV, I think we all agree. Once a day is better than twice a day. And 1 pill a day is better in so many different ways, usually, compared to taking multiple pills at that same dosing. There's some really kind of interesting data looking at large databases that say that there's even less bad outcomes, including hospitalization for reasons I don't completely get. But the selective adherence is a really important thing. Chuck Hicks and I did a really interesting study a few years ago that we didn't really do a good job of explaining to people, but we looked at low and high dose ritonavir taken with another protease inhibitor, and we measured adherence, and we measured drug levels, and we measured all these things, and we found out that a lot of people didn't take their ritonavir. So even though we were comparing, 2 pills versus 1 pill, a lot of people who are on the 2 pills were taking only 1 pill of the ritonavir, or not taking it at all. So there is this disconnect, and I think we're going to embrace single tablet regimens as being sort of an expected thing as more and more of them partner together. Good, yeah.
Yeah, I think that's a great. So the question was about our kind of large treatment-naive studies being underrepresentative both of minorities and women also. And I think that there are a couple of issues, and one is that the NIH and the NIAID has moved away from large comparative trials. And so almost all the large comparative trials are pharmaceutical company trials now. And I think every trial I showed you actually was a pharmaceutical company trial. And they have a goal. Their goal is to get their drug to market before someone else does. So they are going to get a lot of sites and enroll as quickly as possible. And it's just, I don't think it's sexist or racist to say that sometimes it's easier to enroll men than women because men take less responsibility for other people, so they can get hunted clinical trials more easily. And I think it's also somewhat easier for people that have some means. I think race is a marker of means, perhaps, to be in a clinical trial and enroll quickly, which is what happens. And there's no unless the FDA does something, there's no incentive for a company they should just want to enroll as fast as possible. And I think it's a really big problem. So what the FDA tends to do then is to do this post-marketing thing where they get a commitment from the company to do something. And so far, those commitments have really fallen short in terms of actual usable information. But Christine Patterson is actually leading a study here that is going to be the first study that I know of in the US that is specifically only enrolling women. So it's a treatment-naive study that's going to only enroll women. And by definition, that will enroll a large number of women of color. So I think those kind of studies are needed. 2 things are needed. One is either the FDA says that your drug's not going to be approved unless you have studies with X percent of people of color and Y percent of women, or there's more of a commitment towards studies that are really focused on these populations.
I mean, the corollary to that is that the analogous problem is going to occur and continue with respect to prevention because of this low incidence problem. And we're going to be in the same situation of saying, well, how does this work in women? Or, we don't have the information in women because of this low incidence issue that we were talking about a minute And in fact, many of those people will be people of color in the United States.
And so what Ada said is that it will apply to prevention studies also, because the good news, if there's a silver lining, is that most retrospective analyses of multiple studies put together that look, at least by gender or sex, don't seem to see much difference in terms of outcome related to sex. There are a few examples, and David could probably talk about them. In terms of race, there are some differences in outcome, and it seems to be predominantly driven by adherence. And I don't know, David, if you want to add to that.
No, but I think we have to look at, especially with newer drugs, because the differences virologically, they're not there, but in tolerability, we do see some differences that are important, especially with like ritonavir, which is a drug that we still use a lot of and will for a little bit while longer. We don't know about cobicistat. So I think that Christine's project is going to be really, really important. But you're exactly right. It makes us, cringe when we see those demographic data. And we give it back to the companies all the time and point it out, and then kind of hold their feet to the fire and say, I've seen Joe do this and others say, you've got to do a study that looks at this in women. So I think it's really important. I think that's why we did our real study looking at African Americans in Durham, because we really didn't want to answer the question in another population. We wanted to enroll the people who are going to be taking the medicines and look and see what does it do to them. And that was all about tolerability. That wasn't for virologic outcome. I think we need more of those kind of nested studies. So your point is really well taken. It's very good. Yes, back there.
This is a kind of general question.
So the question is about the landscape of affordability in our country, and how much these drugs cost, and are we going to be able to continue to afford them, and what about generics coming out, and how does that change things? You're right. This is a big issue for us, especially in places, for everyone, but especially where there's managed care, whether that be in an HMO or in a state plan or even in our prison system where we're dealing with these formularies. And now we get all this drumbeat of data that show that single tablet regimens are the best thing since seedless watermelon, and then you feel like, well, I can't break up that pill even if it saves us, $5,000 or $6,000 per patient. So there's a lot of forces at work here. I do think that the way the economic trends will dictate to us increasingly what we can and cannot do. And by that I mean, if the economy was to, let's say, take a nosedive again, I don't think we'll have choices as much. I think when the economic forces come down to bear, we won't have the luxury of fighting back and saying, oh, but we need to have the latest single tablet regimen be the preferred one we go to. We won't be able to do that. And just working in a microcosm of our state prison system, we are totally, completely sensitive to their budgetary issues and try to respect that. And we have in the past broken up single tablet regimens so that we can have more people treated. I think right now we're not necessarily in that situation, and our ADAPs are doing a pretty good job, although in some states they are making those decisions. And generics are very tantalizing. But just when that generic comes about, and the others can talk about that, whoa, there's a new formulation that comes out that looks just as good or even better, and there's data that shows that there's less hospitalizations and there's better adherence. So you could see that there's a real big push to try to use the more expensive drugs.
Yeah, the one thing I would add is up till now, and including now, the generics have kind of chipped at the edges of our regimens. Lamivudine, now abacavir. I don't know if a fixed combination of lamivudine and abacavir is actually available as a generic. A fixed dose of zidovudine 3TC is available as a generic, but we really don't use that combination much anymore because I think it really is inferior in terms of tolerability and probably also efficacy. And so when we get one of the kind of anchor drugs, one of those, third drugs that was like on my first slide as a generic, I think then there's going to be a substantial push. And that first drug, as best I can tell, is going to be efavirenz. So there's, as you might imagine, there's like an army of patent lawyers for both Merck and BMS. They make efavirenz, who are trying to find depends on your point of view, loopholes or legitimate reasons that the patent should be extended. You can balance that whatever way you want. Every 6 months that a patent's extended is literally hundreds of millions of dollars. So that's why they do it. So I think when efavirenz becomes generic, whenever that is, whether it's in 2 years or 2.5 years, I think we're really going to see a sea change. And I suspect that we'll be required to give an efavirenz-based therapy as initial therapy. The reason why that one single study, not a single study, but the study called SINGLE is so important, I think, is that it actually showed superiority to an efavirenz-based regimen. And that could potentially be used as an argument to say, no, it's not a non-inferiority issue, it's a superiority issue. So those will be the kind of debate that goes on. And can you give someone a regimen that's statistically inferior but just marginally different? Can you give that inferior regimen in order to save a state or a prison system or a Medicare or Medicaid, literally, hundreds of thousands of dollars. So, I mean, it's a debate everybody should participate in.
It's really cool, though, because I think what Joe's getting at is exactly right. The therapies are so good. We're talking about 83% versus 86%, and now the drugs are starting to catch up, the good drugs. And we're talking about like diminishing returns for any of these newer therapies. They're single tablet, maybe there's some tolerability differences. Someone could argue about the dolutegravir data, well, that was due to tolerability. What about the people who tolerate it? If you tolerate it, you're just as good. So they can push it everywhere. But I see that we're getting closer and closer. We'll have a critical mass of cheaper drugs over the next few years that it's going to be really hard to say that these guys are so inferior to these newcomers that just came out recently or are going to come out. So I'm actually somewhat optimistic that we will have some cheaper options for our people. And if the economy goes great, we'll have the luxury of doing what we've done all this time. But if it doesn't, we're going to be forced to do what Joe talked about. Yeah.
So key, but if you step back to the last one, doesn't really reconcile.
Yeah, so it's a great question. The question is, how do you reconcile the data that shows that there really isn't a difference in risk behavior between black men who have sex with men, and other ethnic groups of men who have sex with men, with what other forces are at work, basically, that describe that, and is it structural, and how do you move forward with prevention efforts? It's a great question. It's a thorny question. I think that the data that has been put out there, in part, I'll give a shameless plug for a paper we just published.
That's so important. It's not shameless. Plug that paper. It's incredibly important.
So it's in the Journal of AIDS, and we looked at a sexual network. So it was a retrospective study that looked at a sexual network of overwhelmingly of young black gay men in North Carolina. And the prevalence of HIV within the network was almost 30%. And so I think the way I put it all together is that the age, and the other things that we saw in the study that are important, is that the age of first appearance in the network dropped over the period of a decade. The age at HIV diagnosis dropped over the period of a decade. And so I think what the way I put it all together was that if you get young men who are ostracized by their community, who don't get safe sex messages in school or at home, and who before, I think that it's not been in research literature, it's in the lay press, this idea that how to be a gay person was an oral tradition that you learned from people who were older than you. And that when AIDS happened in the 80s and 90s, that middle group died. They're all gone. And so there's this age gap between the youngest gay men and older gay men. And that how do you do this, and how do you do it safely, and what does it mean, is gone. That the youngest people are now being dropped into a sexual network with 30% prevalence, and they have no idea how to keep themselves safe. And so it stands to reason to me that if they don't have access to condoms, and they don't have access to health care, and they don't have access to safe sex messages and all of that, that of course they're going to get HIV. That we're doing nothing to address the bigger issues there. And that's why I think in the short term, figuring out PrEP and where its place is in the United States is really important. And if it becomes a bastion of insured white gay men in cities, then it's a phenomenal failure. in my humble opinion.
And if he'd only done a PET scan, you could have
Yeah.
gotten it in the JAMA.
Right.
You know?
Right.
Yeah. Go.
I just needed aortas, and I'd be.
So can I follow up on Perry's question, just because I'm impressed by your answer, and I was also wondering at the same time, the other data you showed and that we've read, just the viral load of the HIV positive men that we're talking about, that 30%, is like, because you showed that African American men who have sex with men are less likely to be on be offered therapy,
Yeah.
be on therapy, and be suppressed. So I'm not going to use the community viral load word, but is the viral load among men who are infected likely not controlled compared to the white networks?
I think that's a really, really important point, and not to continue plugging Greg Millett, but he does really important stuff. He wrote a paper recently that echoed something that our group with Lisa Haito and myself published several years ago now, showing that if you have that men who have sex with an older sex partner had a higher risk of HIV infection. And it makes sense that the youngest group has a low prevalence within that group. As you get older, the prevalence stacks up. And so if you have age mixing, that's a risk factor for infection. That has been repeated now multiple times. And if you get into a situation where now you have a high prevalence population that's not on therapy or is imperfectly on therapy, and so the tier viral load, I won't go community because if Bill Miller is not here, I don't think, so he would have our heads. But that idea that if that tier doesn't have good viral control, and now you're doing age mixing, there's a lot of opportunities for transmission to happen. So a two-pronged approach of getting people into care and retaining them, getting their viral loads down, is going to be critical for long-term prevention, but also identifying the people that are at greatest risk and linking them access to biomedical prevention if they want it, and safe sex messages, condoms, all the things that we know already that work. So it's a big challenge, and nobody has figured that out yet. So, Becky.
Enjoyed your presentation.
So Becky's question is about the undiagnosed people. Maybe a quarter of people with HIV, living with HIV in our country, are not yet diagnosed with HIV. And maybe Christopher can talk to this, because when you looked at the 061 data, you talked about that 11% prevalence at entry into the study. And if I remember correctly, a huge proportion of those people had not known that they were HIV infected.
Well, that 11% was people that were already known. There was 12%.
12%,
so basically the
right,
same number,
that's
but
right.
12% that were newly diagnosed.
Newly diagnosed. I mean, that is extremely tough. It's like if you did a study just trying to find people who didn't know they were HIV infected, you couldn't have designed it almost better than the group of people that were enrolled in 061.
Yeah, I think, so the idea about where are they, and how do you get them tested and engaged, the testing part, I have relatively strong feelings about, and I think people, Joe said no. It's a big shock. I have strong feelings. Home testing is being propagated as an avenue for this. The problem is that we've had home testing since 1996, and the only paper that looked at who was using home testing was published a year after home tests became available. And it was overwhelmingly not people of color. It was overwhelmingly not people at high risk. It was worried well. And so the idea, and that test is still available. You can go to CVS or whatever. It's $80, that test. This summer, OraQuick, well, actually this month, OraQuick, the home OraQuick became available for commercial purchase. It's, I think, $60. So if you're looking at people who are scared of healthcare, that don't want to be engaged, but still want to find out their status, you're pricing it out of the people who are at greatest risk. And so again, not to beat a dead horse, but these are structural issues. If you want to test the people that are at greatest risk, you have to make the test available. And if you have to make it cheaper if you want them to test at home. So one potential avenue is having health departments facilitate home testing, either by sending a kit to people individually or dried blood spots, which is what Mike Hay, hopefully, if and when it gets funded, will look at. is if you send a kit to do a dried blood spot and have centralized testing, will people use it? Is it acceptable or feasible to do it?
Christopher, I've been very impressed, though, by the data we're getting from Washington, D.C. and sort of trickling out from the Bronx with their Get the Test initiatives. These are two very concentrated urban areas with some different sorts of resources than many of the dark, reddish, brick red places you're showing us along the Mississippi River. And I'll tell you, I think that there's so many different answers to this, and there's not one crystal solution. I was just in Missouri the other day, and
you said it right. Yeah.
I did, didn't I? And again, at this little town, and people told me they were just afraid of getting tested. They were afraid of being ostracized. It's the same thing you've heard before. I went to Buffalo, New York 2 weeks before, and the same sort of thing, that people didn't want to talk about it. The leadership of the community didn't want to talk about it. It wasn't being talked about in churches. They were like, we don't want to give a bad name to Buffalo. I mean, so you just see this over and over, some remnants from the past that haven't changed at all, that it is still shocking to me. Whereas a lot of the data from Washington, D.C. and the Bronx is pretty exciting. I just worry that that kind of effort and the resources that are available there that have shown, CD4 cell counts going up, more people getting tested, 5-fold increase in testing. How's it going to reach those dark brown places that are dotting along, the southeast?
One thing that I heard Michael Mugavero talk about is instead of working down the cascade, we should work up the cascade.
That's right.
And we obsess a lot about adherence. But if you look at our clinic, at people that have more than one visit in a year, the proportion suppressed is like 83%. So it's really adherence to care, and we should be working our way up the cascade. So people we've somehow managed to make contact with and get in clinic, we need to keep in clinic. People that have been tested need to get linked. I think that's a more efficient way to address the problem. Not that you should ignore getting people tested, but clearly, as much as the CDC, I think, deserves a lot of credit for saying everybody should have a test, it's not going to work.
It also winds up being a lot of money that's spent on testing people that have zero risk.
Right.
For the sake of saying that you're increasing testing. And the idea of targeted testing, the shift away from that was to destigmatize HIV testing. And yet, by doing that, they've successfully increased the number of tests that were conducted among people that have zero or low risk. Right. And I've since forgotten the number, but if the state statistics for North Carolina...
Double the tests, and only about 20% increase in positive tests.
So if you were getting the right groups, you'd expect essentially kind of a linear increase with the increased number of tests, increased number of diagnoses, and you don't. We've tested way more people, lots and lots more people, and found basically no new infections.
About internet purchasing or ordering of home HIV testing, because to me it seems like the idea you're going to go to CVS and ask, can I have an HIV test for $80, please? It's just ridiculous. But in the type of population that we're talking about, and obviously the cost would still be an issue, I'd assume, via some kind of internet thing. But I would think at least you'd have greater anonymity in the testing.
Yeah. So the question is about internet purchase of home testing and whether or not that might facilitate things. You've been able to buy OraQuick on Amazon for many years, a big kit of them, a big box. So it's been available. It's just sort of flown under the radar. The way that the results are reported for home access, the existing dried blood spot, is by phone or online. I don't know how OraQuick has, you get the result in front of you, and they have sort of support mechanisms online and by phone. But it's a good thought that taking a layer out of it and sort of empowering people to test on their own, I think that a lot of people think that's a good thing. It's just reducing the cost to make it feasible. Yeah.
We're going to have to end because we need to leave this room, but I want to thank everyone for sticking around. I want to thank, again, Michelle and Megan for making this happen, and KJ and everyone else who's helped out, and to our panelists. Thank you.
Yeah, great. Thank you, David.